All right. So, I guess a couple things. I've met with some students now I have several students now regarding the master and his emissary paper, and that is good. Definitely keep that up. And I encourage all of you to make time to talk with me. I can be quite flexible about when I can meet to talk about your master and his emissary papers. I can also do things over email if that's what you would prefer, but I do encourage it. I posted the instructions for the paper way back in the syllabus, the very first day of class. You knew kind of the two different topics for the paper. Um, and I'm definitely happy to help you kind of talk over your paper, or if you have specific questions, I'll even read parts of it and I can give you feedback on it. Don't really want to read the entire paper because I'll be doing that later, and that takes basically an entire week to grade all of the papers. So I don't want to read all of it now, but I definitely, if you have specific sections or questions about the paper that you have written, I'm happy to answer that and give feedback on specific aspects of your paper. So that's that. Also, quizzes. I'll just remind you the way things will be going for the rest of the semester. There's a couple more quizzes left, 2 or 3 more, I believe. And I will be sending out emails when those are ready to go, announcing the due date. And all of the due dates for those quizzes at this point will be after the dates that they were originally scheduled to be due. Got a little bit behind on the quizzes. So for all quizzes moving forward, I will send out emails saying that they're posted on Blackboard and announcing the due date. All right, so the rest of today is just finishing up, hopefully, this memory chapter. We've talked about sensory memory. We talked about short-term memory. And last time we did start talking about long-term memory, and that's this brain that we're looking at here has some regions that here in the middle temporal lobe, which are kind of a focus of the chapter in your book, the medial temporal lobe structures, which are important for long-term memory. And the most important of which that we'll talk about in this chapter, the hippocampus, pararhinal cortex, entorhinal cortex, parahippocampal cortex. Really the most important one out of those four, is the hippocampus, though these 3 other regions, pararhinal cortex, entorhinal cortex, and parahippocampal cortex, all are thought to work with the hippocampus for the purpose of long-term memory. So this is the structure we'll be kind of talking about today, but I kind of want to back up just a little bit and remind us of this difference between declarative versus non-declarative memory, because that brain structure we're just looking at, the hippocampus and those surrounding structures, they happen to be really important for declarative memory, but the evidence suggests that non-declarative memory does not involve those medial temporal lobe structures. So I want to make sure we understand before we go into talking about those brain regions, the difference between declarative and non-declarative memory. Declarative memory I mentioned last time is memory that can be verbalized, stated as facts. And there's some examples shown on the slides right now. And then contrasting that with non-declarative memory, these are things that cannot as easily be stated verbally or as facts. So really the best examples are like actions and procedures, like learning how to play a musical instrument, learning how to do some type of a move in sports, like serving a tennis ball or a golf swing. Another example is eye blink conditioning that actually is considered a type of non-declarative memory. It's kind of just a reflex that your body does. We talked about last time, and then in the previous chapter on motor system, puff of air blows into your eyes, you will reflexively blink. But if that puff of air, you can learn an association between that puff of air and a tone, such that eventually the tone by itself will cause your eye to blink. So that's a form of long-term memory, but it's not one that you necessarily verbalize. It's something that your body seems to know, and it does. And that's kind of the nature of the memory. It's less about the facts that you're able to state and more about kind of the reflexive things that your motor system, your body does. And that's the general nature of non-declarative memory. We haven't really talked too much about implicit associations. We will get to those, I believe. We talk about that in the language chapter, which is another type of non-declarative memory. But for now, really the main types are just actions, procedures, and classical conditioning. Those are the main types of they're long-term memories, but they're not memories that you verbalize. They're kind of like reflexes or things that your body can do, but that you definitely are acquiring some type of knowledge over time. So they are long-term memories. So now this takes us to patient HM, a pretty famous patient who was having seizures. A common reason to have brain surgery is you're having seizures, and those seizures are coming from somewhere in the brain, and doctors can remove parts of the brain potentially. They don't, obviously there's some parts of the brain you can't remove, but there are some parts of the brain which you can remove, and they will have relatively minor effects. And it turns out the hippocampus, it's a region of the brain that you can have it removed and you'll still survive. But this patient, HM, he had his hippocampus removed. And it was the very first time that this type of a procedure had been done to remove these structures in the medial temporal lobe. I showed you pictures earlier. The hippocampus, I'll show it to you real quick. Here's a kind of a side view. The hippocampus is this thing right here. Notice that we're in the temporal lobe right here. The hippocampus is this part right here. The amygdala, which is a region, by the way, we'll be talking about in chapter 10 on emotion. That part of that region was removed as well. So it's kind of like a big swath of the medial temporal lobe, but definitely a big thing that was removed was the hippocampus. But I just want to point out that with patient HM, the hippocampus was not the only region that was removed. The hippocampus definitely was removed bilaterally, both hemispheres, but so were a couple other brain regions as well in the medial temporal lobe. As I said, this is kind of the first time that this type of a surgical procedure had been done to remove such large portions of the medial temporal lobe, especially the hippocampus. And so the doctor who did this, who my understanding is the doctor performed this surgery, kind of had a reputation for doing risky surgeries. Kind of liked the science of doing these kind of never before done procedures. And apparently he kind of talked patient HM into doing this procedure. And it turns out to have had, although as I mentioned, HM survived, like he was still able, his heart was still beating, he was still able to breathe. He had major cognitive impairments after this procedure. Basically, he lost his ability to form new long-term declarative memories. So that's what this is right here. The main problem that he had was anterograde amnesia. Anterograde amnesia basically means that after the surgery, he woke up out of the surgery, and the main problem he had was he could not learn new things. So a common type of declarative memory would be like you meet a person and you say your name, and then you might have a small conversation, but then you'll see that person maybe a couple days later and you remember that person's name. You see the person, you say, hey, so-and-so, and you remember their name. That's a type of verbalized memory. You're remembering a person's name and you're able to say their name. It's kind of like a fact. That person's name is, whatever. Patient HM wasn't able to form new long-term declarative memory. So if you met him, he actually could remember your name using a short-term memory. So you could say, hi, my name is Matt. And he'd say, hey, Matt, how are you doing? And you could talk to him, and 30 seconds later, you'd be like, all right. He could say like, okay, see you later, Matt. But then if you met him the next day, the short-term memory, that's too long for short-term memory to help out. By the next day, he would have forgotten that my name was Matt. So his short-term memory was intact, but his able to encode information into long-term memory was not working. He also had slight retrograde amnesia. So what this means is that he did lose a little bit of the memory of some of the stuff that had happened to him just before the surgery. And so that's an important aspect of saying it was slight retrograde amnesia. For instance, he still remembered stuff from a long time ago. Like he still remembered his childhood. He remembered when he was like a young adult, even a middle-aged adult. He's pretty old to my understanding. So he remembered things from, definitely from years ago and even maybe 1 or 2 years ago. But the stuff that had been recent, but right before the surgery, like a couple months up to the surgery, he forgot all of that stuff or most of that stuff. So once again, he lost a little bit of the memories of stuff that happened in the recent past, but his memories of the distant past were quite intact, though his main problem was the inability to form new long-term declarative, that is verbalized, factual memories. So that's a lesion study, as case study. There's also been some imaging studies now that basically suggest that out of those regions of the middle temporal lobe that were removed in patient HM, probably the one that was most important for his impairment, his anterograde amnesia, was the hippocampus. This is a region of the brain. I'll show you some evidence here in a second. That is pretty important when you're encoding information into your long-term memory. And if you can't encode that information into your long-term memory, then that information is never going to get to your long-term memory, and then you won't be able to retrieve it later on. So yeah, now I'll kind of talk more about, I'll just say one more time, this is the hippocampus right there, and it works in concert with these other brain regions around it, probably even of the amygdala, which we'll talk a little bit about in chapter 10. But these regions, parahippocampal cortex, entorhinal, pararhinal cortex, these regions all work with the hippocampus to encode new long-term declarative memories. And we'll be talking about a little bit about how these brain regions work with the hippocampus to encode long-term declarative memories right here, in fact. So this is a little bit of a complicated diagram, but you don't have to know everything about this. If this were a graduate level course, for instance, I would definitely want the students to know all of the details about these substructures within the hippocampus, dentate gyrus, subiculum, if I'm recalling that name, CA1, CA3. But you guys don't have to know these. Just know that basically this part right here, this is the hippocampus in this diagram, and it is composed of these substructures. Do I have other pictures? Yeah, here's another picture of the hippocampus, and you can see some of those substructures within it right there. Once again, you're not going to have to know anatomically any of those substructures within the hippocampus, but in this diagram, this is the hippocampus right here. And one of the theories, which is presented at least in your textbook, and it's a theory which is out there in the literature, is that the way information, there's a kind of like two different pathways that lead in to the hippocampus for encoding long-term declarative memories. One of the things I kind of like about this theory is it kind of has a similar distinction between the two different types of inputs that we talked about way back in, what was it, chapter 5, the what and where, that is the ventral and dorsal pathways of the brain. Remember, the ventral pathway was thought to represent information about objects, and it helps you recognize objects, whereas the dorsal pathway was important for representing where in space various objects are, so you can act towards them. And there's kind of a similar distinction here with respect to the inputs into the hippocampus. You can see that here there's kind of a what input according to this theory, according to this model, and there's also a where input. And so the what input would be, I'm forming a long-term memory about this object, or I'm forming a long-term memory about this person. That is the what. The thing, which is kind of the memory, is about some object or some agent, something. That is the what. And it's thought to involve PRC. That is the pararhinal cortex. So I'll show you that real quick. The pararhinal cortex right here, PRC, is this. This is thought to be the region of the brain which inputs what information into the hippocampus. And you can see how these two regions of the brain, PRC, pararhinal cortex, is right there next to the hippocampus. And the idea is that pararhinal cortex will input what information? Information about the objects that you are trying to form long-term memories about. Contrasting that here with the parahippocampal cortex, which is thought to input where information, but I'm going to say it's a little bit more general than just where information. It's more like contextual information. And this is a pretty important addition to this. It's not just about the spatial arrangements of things per se. Where is like the contextual information. So I'm forming a memory about so-and-so. The where aspect of it, let's say I meet a person and then I remember their name. That's like I'm remembering information about a person. That's the person is the what. The where information would be like, where did I meet the person? So I have this memory of someone and I know their name because of this event that occurred in the past where I met the person, and then I remember their name as a result of that. But part of the thing that you remember isn't just the person's name, it's also kind of the contextual information that occurred when you learn that person's name. So that's what's considered where here. And so this is the parahippocampal cortex. Let's go back to this thing. That's this part right here, parahippocampal cortex, pararhinal cortex. Those contribute what versus where information into the hippocampus. And then the hippocampus can eventually form long-term memories. But they do so kind of through an intermediary brain region, EC. This is the entorhinal cortex right here. which is thought possibly to kind of bind or combine these different types of information into a single representation. So I want to go back to our little brain here, and then a schematic possible kind of way that the information is traveling here, although this is still quite theoretical, hypothetical, is that information for the pararhinal cortex is going here to the entorhinal cortex. Information for the pararhinal cortex is also going here to the entorhinal cortex, and then they're kind of pushed up into the hippocampus right here. So there's like kind of these two pathways that are converging on the entorhinal cortex, one from pararhinal, one from parahippocampal cortex. They kind of converge at the entorhinal cortex here, and then it sends it up into the hippocampus. And then the hippocampus does some things which allow long-term memories, presumably, to be formed, long-term declarative memories. So here is another, this is a close-up, an anatomical close-up of the hippocampal formation. It shows a little bit of the entorhinal cortex right here. And you can see that it's got that input. Once again, the entorhinal cortex, the EC, is thought to be the input into the hippocampus. And then you can see that there's, all these loops and arrows within the hippocampus, which are thought to kind of show the direction of information traveling within the hippocampus. Once again, this is a pretty advanced map right here. So I'm not going to require you to know the specific anatomical details about anything really which is going on within the hippocampus, but just notice there's all these different substructures within the hippocampus. Information is traveling among them. And then there is this interesting kind of feedback loop where information is entering the hippocampus, and then there's actually an output from the hippocampus back into the entorhinal cortex. So there's kind of this like loop that's going right here. And this is taken from an article in Current Biology, which is a pretty good journal. It's got a really high impact factor. And it's a general biology journal, so it's not specifically a neuroscience journal. They'll publish things from biology and other fields outside of psychology and neuroscience. But here in this journal, they referred to this kind of configuration here, the trisynaptic loop. The loop represents the fact that there's kind of this feedback loop. Information enters the hippocampus via the entorhinal cortex, and then information comes back to the entorhinal cortex. Now, that's not all that's going on. There's probably, in fact, there's pretty clearly some evidence that there are projections away from the hippocampus as well, but we're just zooming in right here on just kind of the hippocampal formation and the entorhinal cortex. There's that feedback loop right there. Information enters and it comes back to the entorhinal cortex. So here's kind of like a summary of some of the proposed functions of these different brain regions that I've been talking about. The entorhinal cortex, as I mentioned, it seems to be taking in information from both the pararhinal cortex, which does what, that is object information, and the parahippocampal cortex, which does where, spatial, contextual information, and then it sends those up into the hippocampus. So that's why it says here entorhinal cortex does spatial and object information, because it's taking information from those two different pathways and they appear to be combined there. So one thing that's been proposed is maybe this is a region of the brain which is kind of binding these two separate pathways into one before they are sent up into the hippocampus. That's a hypothesis, at least. The pararhinal cortex, as I mentioned before, it inputs object information, so information about the what. And something which has been well studied is the fact that the pararhinal cortex responds highly to familiar things. So when you see something which is familiar, the pararhinal cortex becomes more activated. Let's see, and then finally, parahippocampal cortex. I've mentioned it inputs where, it inputs spatial information. In general, it inputs contextual information. I have a study about contextual information here. So this is an example provided from the textbook about the type of information that the parahippocampal area appears to be contributing to the hippocampus. And it's talking about contextual information. I just want to point out in this particular study, the parahippocampal cortex is not inputting spatial information per se. It's not inputting information about where. It's inputting a different type of contextual information. And so I'll be talking about that very shortly. First, I want to talk a little bit about the hippocampus and some of these structures around it as far as the type of information they appear to be representing. So we're really looking into the details now of some of the stuff that the hippocampus does. And to be perfectly honest, like the relationship between these findings and the formation of long-term memories, which the hippocampus clearly does, is a little bit, it's not exactly easy to see the connection. There probably is one. Maybe in the future, the connection will be well known to scientists. But right now, there's kind of these isolated facts about the hippocampus. We know it does this. We know it does this. But there hasn't necessarily been a cohesive theory which is able to explain all of the different aspects of the hippocampus and how they're all involved in encoding long-term memories. So place cells are a thing which are shown in the hippocampus. And this is a place cell right here. So this is a little bit of a complicated diagram. This is taken from another article. I believe this is like a Nature Neuroscience article, so another high impact journal. And all of this, everything in A, this represents a single neuron in the hippocampus, one neuron. It is a place cell. Place cell, that's what this is, a place neuron or a place cell. And these red dots represent action potentials. But you notice that there's a lot of red dots here, but there's not a lot of red dots over here. Overall, the square formation of this neuron, like the square that you see here, represents an animal, probably a rat, that is moving within a square environment. And that's what these little black squiggly lines are. This is an animal moving around an environment. And these red dots, once again, these represent action potentials that are fired in that animal's hippocampus, a single neuron. And one of the things you notice is that when the animal is over here, there's not potentials being fired. When the animal is over here, there's not a lot of action potentials being fired. But then when the animal moves to this one specific location in this environment, it's like a square environment, like a room, like I'm in a square room right now. When the animal's standing in this specific part of the room, it causes this one neuron in the hippocampus to fire action potentials vigorously. A lot of action potentials are fired there. So clearly, the firing of action potentials in the hippocampus are representing some location in space. And there's lots of them. It says they're located throughout the hippocampus. There are a lot of place cells within the hippocampus. Basically, there are a whole bunch of neurons in the hippocampus, and the job of those neurons appears to be representing a specific location in space within some type of contextual environment. So those are place cells. Then there's another type of neuron. These tend to be located in entorhinal cortex. So remember, entorhinal cortex is the region which inputs into hippocampus. The hippocampus is where that place cells are located. Place cell is basically a neuron which responds to one specific point in the environment by firing lots of action potentials. That's the hippocampus. There's also the entorhinal cortex, which inputs into the hippocampus. And this type of a neuron is called a grid cell, and it is located not throughout the entire entorhinal cortex, but that medial part of the entorhinal cortex. And grid cells have some interesting, and I don't want to say confusing, but they're kind of like, what are they doing? That's like the main question about grid cells. They have some interesting properties, but it's not 100% clear what exactly the purpose of this is, at least to me. So the property is, it says right here, you have a grid-like pattern of firing locations. So they're kind of, once again, this whole thing, all of these red dots represent the firing of a single neuron. This whole thing, all of the red dots in this B diagram represent action potentials of just one neuron in medial entorhinal cortex. And once again, these black lines represent an animal moving through this square environment. And the thing which is interesting about grid cells is that, there are clearly places when the animal is walking that the grid cell does not fire action potentials. But then there are places that when the animal is standing in that position, these neurons do fire action potentials, like right here. So imagine an animal is kind of walking against the wall of a room. It starts in the corner, this one neuron, entorhinal cortex, not firing action potentials, but then it walks against the wall for a little bit to right here, and then that neuron starts firing an action potential. It continues walking along the wall here, then it stops firing an action potential for several steps. And then the animal gets here, and it starts firing an action potential again. And then the animal walks this way, it stops firing action potential, walking this way, and then eventually starts firing an action potential again for a few steps, then it stops. And you can see that there's kind of this intricate pattern, and it forms a grid. That's why they're called grid cells. So what's different about grid cells and place cells? A place cell responds to only one place in the environment. A place cell is a neuron, which responds to only one place in the environment typically. Whereas a grid cell is a neuron which fires action potentials in a bunch of different places when the animal's in a bunch of different places in the environment. And the environmental places that cause this neuron to fire action potentials are organized kind of like a grid. So as I said, the significance of this for forming long-term memories, it's not entirely clear to me. Maybe people that are, I imagine there probably are some theories about this, about exactly what they're doing. They're definitely not talked about in a lot of the cognitive neuroscience textbooks that I've looked at. What is going on here? Why does it do this? I will just say that an interesting aspect of the entorhinal cortex is it has this recurrent processing. And that has to do with this figure right here, entorhinal cortex right here. This is where those grid cells are. The place cells are in here. The entorhinal cortex inputs information, you know, spatial and object information is inputted into the hippocampus right here. There's some type of information processing is going on in the hippocampus, but then there is a pathway that leads back to the entorhinal cortex. And that's what I mean by recurrent. You have like an input in and then an output back into the same region. So it's like a loop. That's what is meant by recurrent processing. Possibly the reason why entorhinal cortex, some of the entorhinal cortex neurons have this interesting property of grid cells has something to do with the fact that they're both inputting into the hippocampus, which has space, you know, which represents space through place cells, but then that information is being fed back into the entorhinal cortex. And this might represent kind of this information that is in a loop, a feedback loop, or this recurrent processing. That is definitely just a hypothesis, though. Placing grid cells. So it's pretty clear that that in the hippocampus and the medial temporal lobe, information about spatial location is being represented. As I said, place cells are kind of straightforward. One place in the environment, the animal standing, the neuron fires an action potential. Grid cells, a little bit more complicated, but no matter what, when you're looking at these two types of neurons in the medial temporal lobe, it's pretty clear that one of the things that the hippocampus is important for is remembering spatial arrangements of things. And so this has been studied in animals. I'm pretty sure this is not in your textbook, but this has been studied with animals. They've been studying the importance of spatial information with respect to the hippocampus, and they've done it with animals. So they'll actually do studies where they'll remove the hippocampus in animals, and they'll see how they perform in this task called the Morris water maze. So the Morris water maze, they'll take a rat and they'll put it in this, you know, bucket of water. The bucket of water is definitely deeper than the rat can stand on. So it's the water is deep, so it's not the shallow end. The animal cannot just stand up and chill and you know have its and not be under. Like it has to swim constantly when it's in this water, with the exception of this little platform right here. So most animals, but definitely rats, do not like being in water that is really, really high, you know, that's really deep, because they don't like to have swimming constantly. It takes a lot of energy to swim, and obviously they don't want to drown. And so when the rat is highly motivated to find this platform, and so they will, if you notice these things on the wall right here, they have these, I guess you could call them markers, and they're like spatial locations in the environment. And the animal, in theory, can use these markers to help know where the platform is. So as I said, the animal is highly motivated to know where this platform is because he wants to get there as soon as possible. Because if it stands on the platform, it's like, this is like the shallow end right here. The animal will be a little bit wet, but the animal doesn't have to swim constantly. They can just kind of stand there on the platform, and then they won't have to be using energy to, you know, survive. So highly motivated to find this. And the animal can use these kind of landmarks, I guess you could say, to help the animal know where this platform is. For instance, if you look at this, the animal could be like, oh, the platform is right in front of this, and it's close to the blue square which is on the wall right there. So if the animal is kind of aware of the spatial environment, if it's able to kind of encode this stuff into long-term memory after doing this over and over and over again, the animal can start to learn, oh, if I want to get out of this pool, I don't want to just swim around randomly. That's kind of what appears to be going on here. The animal, the first time you put them in the water, they don't know where that the platform is, and so they'll kind of just swim around looking, you know, for something to save them. Eventually, they'll find the platform. But then after a while, they might hopefully learn, oh, if I want to get to the platform quickly, there's this landmark here. I can look at that landmark and then swim over into the direction of that landmark, and I'll have better luck finding the platform if I'm swimming around the location which is close to this blue square. But note that that takes that requires a long-term memory, and it requires encoding information about where things are in relationship to each other. The platform is located in the relative proximity to the blue square, and it's kind of in between the blue square and the red circle. And so you can see after learning, after an average rat, or rats whose hippocampus is working, when you put them into the environment, the Morris water maze, they tend to swim pretty quickly to the platform. But they've studied, as I mentioned, the hippocampus and how that is involved with these types of long-term memory. So I guess one thing I should mention is it's kind of interesting that we're studying the hippocampus, which is thought to be involved with declarative memory. That is, memories that you can verbalize, but we're studying rats, which are animals definitely that cannot verbalize memories. But presumably this is kind of like the precursor of the types of memories that we could verbalize. We could say, for instance, even though a rat can't say it, we could say verbally, the platform is located close to the blue square. The rats can't say that, but it's thought to be kind of like the precursor knowledge of a declarative memory. And the researchers have been studying these types of long-term memories in rats. And they've noticed that rats with hippocampus lesions can swim straight to the platform when they're dropped in the same location in the Morris water maze every time. So this might seem a little bit weird. I just said that the hippocampus is important for long-term memories, and now I'm saying that these rats with hippocampus lesions can swim straight to the platform quickly, but there's a catch here, and the theory is that if you put a rat in the exact same spot in the Morris water maze, maybe I should show it here real quick. If you put the rat in the exact same spot, the rat can learn using procedural, that is non-declarative memory. Oh, when I'm dropped here, I need to swim straight to the left real quick, and then I'll get to the platform. If it's dropped in the same position every time, the rat can form a procedural memory, which will allow it to get to the platform quickly. And rats with hippocampus damage, their procedural memory, their non-declarative memory, their action memory, that still works. And so you can drop them in the same location, and rats with hippocampus lesion, they'll get to that platform quickly because they can just do a procedural memory. Swim to the left swim to the left until I find the platform, and then I'll be good. That's a procedural memory. Rats can do that. However, if you drop the rat from different locations, so sometimes you put it over here, the rat, when you put it into the pool, sometimes you put it over here into the Morris water maze, you put it in these different locations. Rats when you're put in different locations every time, if you want to find the platform quickly, you have to use those landmarks that are on the wall. You have to be like, oh yeah, the platform was close to the square. The square is over there. I should swim over towards the square, and then the platform will be good. Rats with hippocampus lesions can't do that. And so this is a good evidence. This is kind of like more evidence that the hippocampus is important for some types of memory, those resembling declarative memory. In this study, learning the associations between the location of the platform and the shapes on the wall. The animal can't do that very well when the hippocampus is damaged. But once again, the animal still does seem to be able to form procedural memories. So this is once again evidence of this dissociation. Some types of memories require the hippocampus. Some types of memories don't require the hippocampus. And procedural memories are kind of, one of the main regions which is thought to be important. Procedural memories would be like the cerebellum, whereas other types of memories clearly involve the hippocampus, the medial temporal lobe structures. Okay, so now this is a imaging study, a neuroimaging study. And this is one, is obviously using human beings as participants, which is forms the basis for kind of some of these suggestions regarding what some of the substructures in the medial temporal lobe are doing. In particular, the parahippocampal cortex, the parahippocampal gyrus cortex. I'll remind you, go back here. PHC, that's parahippocampal cortex. This is the region of the brain that is thought to input into the hippocampus, entorhinal cortex and the hippocampus, where information, but I've mentioned it's oftentimes called where, but you should think of this more generally as contextual information. It's not just information about where something is located, but it's basically information about the environment, and where is one part of the environment, but it's just contextual information in general. And the parahippocampal cortex, based on this study, seems to be important for encoding part of this contextual information. So I'll just point out, this is a study. It will definitely be on test 2. And it's a study which students, I've learned, some get it, but this is a hard question every single time this test occurs for the students. In the past, I've actually literally included this figure in the test. I would probably do that again for test 2 when we get there. So just some heads up, this question will almost definitely be on test 2. And the way the study works is there was 2 phases of this experiment. There was an encoding phase where people would see these images right here. And so what they were supposed to encode was basically, well, actually participants weren't really given that specific instructions about to remember anything at all. They weren't like necessarily trying to remember, but they were asked some type of questions. I don't actually remember the questions. In the encoding phase, though, they basically just wanted people to look at these pictures. And they assumed that people would encode some information about these, I call them pictures, I guess it's like these words or these images right here. And then while this was occurring, they were measuring brain activity during the encoding phase. But what the researchers were interested in, like later on, there was a recollection, or you could say a retrieval phase. And this is the phase where the researchers, what they did was they would present things to the participant, and those things would be words. And some of the words were words that they had seen during the encoding phase. So like during the recollection, the retrieval phase, the researchers might show a participant, here's the word nickel. Did you see this word before? Well, actually, the specific question was, how confident are you that you've seen this before? And it's a scale of 1 to 6. Definitely old means, yeah, I have definitely seen that word before. I saw that word for sure during the encoding phase, that earlier phase of this experiment. If the participant gave a 1, that means, no, I've never seen that word in this experiment before. This is a new word to me for this experiment. Never seen it before. So that's the first thing. On the second phase of the experiment, the retrieval phase, the first question participants are asked is, how confident are you that you've seen a given word before? The word nickel, they should give an answer like 6. I've definitely seen that word before, because there's the word nickel right there. But then if you show them the word bottle, they didn't see that before, they should give an answer like 1 or 2. No, this is a new word. I haven't seen this in the experiment yet. So that's the first thing. They were judging the familiarity of words during the second phase of the experiment. Some of those words they had seen before in the first phase of the experiment. Some of those words they had not seen before. So that's like part 1. The second part, though, is really important for understanding the parahippocampal cortex. And that's this part right here, reporting the correct color. So for those words that the participant said, yeah, I've seen this before, nickel, I definitely remember seeing that earlier during the encoding phase. And then the researcher would be like, oh yeah, okay, so if you did see that word, what color was it presented in? That is the source judgment. That is a term from the textbook, the source judgment. It's basically information about the context in which some memory was formed. So you formed a memory, a couple types of, a couple aspects of this. The object of the memory is the word itself. I have a long-term memory of seeing the word nickel during the encoding phase. That's part of it. That's the what. the word itself. But then there's aspects of the environment or of the context that were also encoded into long-term memory. And that contextual information was, what color was the word when I saw it? The word nickel was presented in green, whereas another word that was presented during the encoding phase, deer, that was presented red. And so for each word that the participant says, yes, I definitely saw that in phase 1, I definitely saw that in the encoding phase, the researchers would ask them a second question, which is assessing a source judgment. And that question was, what color was the word when you saw it in phase 1 during the encoding phase? And then here, it's kind of complicated brain diagrams. But the most important thing is that they do show parahippocampal activation. And so for the book uses some terminology here, which makes it a little bit confusing. A correct recollection. That's what these are. These are brain regions that were basically activated when participants performed correct recollections. To help you understand what I mean by that, I'll go back to this slide real quick. A correct recollection means that during the second phase of the experiment, you judged a word as you've seen it before. So it's a word that you saw before and you said it was a 6, you definitely saw it before, and you got the color right. And so for those words, remember brain activity was measured during the encoding phase. So the first time you saw these words, they were measuring your brain activity. Those words that were later correctly recollected by the participants, those words that were judged correctly as I've seen them before, and the color was correctly reported, those words strongly activated these 2 regions of the brain during encoding. So that's one of the things I think makes the study complicated, is they were, in this particular one, they were measuring brain activity during encoding. And they were kind of using participants' behavioral performance during the second phase, retrieval, to kind of help to kind of decide how to analyze the brain data. Those words that participants were able to correctly recall, they say they were familiar and they got the color right. When people were originally encoding those words, these were the regions of the brain that were highly activated. And as a result of these, the nature of that source judgment, it's contextual information. It's information about the object, which is the word, and then the kind of the context in which that object was embedded. They show this activation in parahippocampal cortex, which supports this idea that the parahippocampal cortex is important for encoding contextual information. The mnemonic device I kind of think about it is, you remember a person, like the person is like, you remember a person, and that's like the object, that's the what. But you also remember like what color dress that person was wearing, or what color shirt that person was wearing the first time you met them. So the object, the thing you are remembering, is the word, which is kind of like a person. In this study, though, the words could be different colors. That's kind of like remembering what color shirt or what color dress a person was wearing the very first time you met them. At least that's how I kind of think about it. Maybe that's helpful for you as well. Parahippocampal cortex is important, therefore, for encoding contextual information or a source judgment, the information about the color that the word was originally presented in during the encoding phase. So that is a tough one. I might review that study. I might review that study one more time before we completely, next time I might review that one more time before we get to chapter 10. Okay, so one last thing in this chapter, and then we'll call it a day, which is consolidation theory, which is, I don't know, I think it might be one of my favorite things about the memory. I find this theory to be very interesting. And yeah, I remember reading about it in the graduate school in my systems neuroscience class, and I've been interested in it ever since. And then this is a very appropriate time to be talking about consolidation theory in this chapter on the hippocampus, because the hippocampus is thought to be one of the primary structures involved in consolidation theory. So we already know the hippocampus is involved with encoding long-term memory. But it actually turns out that the hippocampus is involved with retrieval of newly formed memories as well. So this is the hippocampus can do a lot of stuff. It's clear from HM, patients like HM, if your hippocampus is damaged, your ability to form new long-term declarative memories is definitely going to be impaired. But it turns out that The hippocampus is also important for retrieving some types of memories. So if I just formed a long-term memory like a week ago, and then today I'm retrieving that memory from a week ago, it turns out that when I'm retrieving that information, from a week ago, one of the brain structures that I'm recruiting is my hippocampus. But that only happens with newly formed memories. And it says here, the involvement of the hippocampus actually the involvement of the hippocampus in retrieving something declines the further away into the past that memory was formed. And this kind of makes sense because if you think about HM, he did have that slight retrograde amnesia. Retrograde amnesia meaning some of the stuff that happened right before the surgery, he actually forgot. His main problem was forming long-term declarative memories after the surgery. But he also forgot a few of the things that happened right before the surgery, couple weeks right before the surgery. That stuff he seemed to have forgotten a lot. And presumably it's because his hippocampus was damaged. And those recent memories that he had just formed, you know, the couple weeks before the surgery, his hippocampus was gone, and you need your hippocampus to retrieve those memories when they're freshly formed, when they're newly minted memories. You need your hippocampus to retrieve them. Those memories from your childhood, though, those things are so long, so into the past that you don't actually need your hippocampus to retrieve them anymore. You can use other brain regions to retrieve those memories. You don't need the hippocampus. And this is kind of getting at consolidation theory. The idea is that at first, you need your hippocampus to retrieve freshly formed memories. But then after a while, that memory becomes consolidated. And what consolidated basically means is other cortical regions. So, you know, regions out here, the hippocampus is kind of in the middle of the temporal lobe. But then after a while, those memories have kind of consolidated and they've moved to other regions of the brain. Usually the theory is that they kind of move to outer cortical regions, which is why I keep doing this with my hand. They move from kind of to the medial temporal lobe, the middle of the temporal lobe, more towards the outskirts of the brain. At least that's the main theory that I've heard about in consolidation theory. And so this is consolidation, the movement away from relying on the hippocampus for retrieval and instead relying on these other outer cortical regions that become more important for retrieval with time. So here's some evidence. I have some evidence for this, for consolidation theory, which comes from fear conditioning. So this is a contextual environment, like a cage for an animal. Starts to be, and it uses the word conditioning, but this is probably not considered a form of classical conditioning. It's probably not considered classical conditioning. But fear conditioning, basically, there's different cages or different environments that an animal can be placed in. And one of those environments, the animal, every time it goes into the environment, something bad happens, like it gets shocked. And so the animal starts to learn, oh, I do not want to go into that environment. If I do, bad things are going to happen, like I'm going to get shocked. And the way you can see evidence that animals have learned about this environment being bad or causing them pain is that animals will show freezing behavior when they're later placed in the context that had previously shocked them. So there's all these different cages with all these different contexts the animal can be placed in. The one context that had shocked the animal previously, if you put the animal in that again, they basically go in there and they won't move. Presumably they feel like freezing is a way of protecting themselves. If they walk around, they're likely to experience the shock, and they freeze as a way of trying to protect themselves. They've learned, oh, this environment is not good. This is not a good place to be. I'm going to protect myself by not moving at all. So freezing behavior, a lack of moving, indicates that the animal appears to have learned or remembered this environment. This is a bad place. And so, yeah, this image is a little bit blurry, but this was taken from an article where they were kind of looking at fear conditioning in rats. And the dependent variable here is freezing behavior. So basically this means is if the bar is high here, that means when the animal is placed in an environment, they stop, they don't move at all. But if the animal, like this low bar right here, this means that the animal was not freezing that much, that the animal was locomoting, was moving around more. So the tall bar means, yeah, the animal remembers that a given environment shocked than previously. A lot of freezing. The animal appears to remember that the environment's bad. Right here, though, the animal is not freezing very much, and the animal appears to have forgotten that this environment caused them pain previously. And so there's some important things going on down here to help the idea, to support the idea that this is an example of consolidation. So you notice right here, it says 1D. So this is right here, they're measuring the behavior of the animal 1 day after training. So on day 1, the very first day of the experiment, the animal goes into the different environments, different contexts. One of them will cause a shock. Then after that, on the beginning of day 2, which is where these data are collected, so 1 day after, the very first day, that's what this is, 1 day after training, some of the rats will be given a saline injection, which is basically placebo. The other half of the rats will be given this. I actually don't know what that acronym stands for. What I do understand is this is an injection which is meant to reduce the activity of the hippocampus. That's what the injection of medicine basically into the animals, which reduces the activity of the hippocampus. And if you do this right before you put them into you give them this injection on day 2, 1 day after training. And basically what you do is you turn off their hippocampus. Remember, I said, for those newly, freshly formed memories, the hippocampus is important for retrieval. In this situation, if the animal cannot remember something from yesterday, what that means is the animal is going to move around a lot because they're not remembering that this specific context causes them pain, and so they'll move around a lot. And that's what's happening here. When the animal's hippocampus is deactivated by this medicine right here, they basically are not able to retrieve what they learned yesterday. This environment bad. Therefore, they're more likely to move around. They show low freezing behavior. Whereas the animals that receive the placebo, they know to freeze. They remember, yesterday I learned that this environment's bad. The animals that on the, right before testing on this day, hippocampus was deactivated. They were no longer able to retrieve that information from yesterday. But the really crucial finding comes from what happens on day 28. So this is, 28 days after the initial training. 28 days after the initial training. And then the rats, half of them are given saline. And then the other half of the rats are given that medicine, once again, which deactivates, or that drug, which deactivates the hippocampus. And if you look, there's not a significant difference between the freezing behavior of these two groups. The rats that received saline, their hippocampus is fine. They appear to be able to remember that 28 days before they had learned that this environment was bad and they're going to get shocked. And so they show pretty high freezing behavior. It is notable. They show a little bit less freezing behavior than they did when they were tested just 1 day after training. So they seem to have forgotten a little bit over the course of those 28 days, but it's 28 days. So they forgot a little bit about the environment causing them a shock after 28 days. But you can see their freezing behavior is still relatively high. And then, but the most important thing is you see these rats right here. These rats on day 28 days after the training, these rats still show freezing behavior. Their hippocampus is deactivated 28 days later, but it doesn't matter, apparently, because as I mentioned in consolidation theory, your hippocampus is important for retrieval when that memory is freshly formed, as it was in these rats. When their hippocampus was not working, they couldn't retrieve the information, this environment is bad. But 28 days later, this allows time for consolidation. These rats, their hippocampus is impaired because of the drug, but it doesn't matter because through 28 days, presumably, that memory information has consolidated. It's moved around to other brain regions, which now have picked up the slack. They don't need the hippocampus to retrieve the memory of what happened during the training earlier on in the experiment. They can use other brain regions, and so they show the same amount of freezing behavior as the rats in the placebo group. So the only difference, this is a significant difference right here, occurs day 1 when the memory was freshly formed. Day 28, there is no difference, and this provides evidence of consolidation. So last slide here, and then I know this has been kind of a long lecture today. This is kind of an interesting, this is taken from, I think another Nature article as well, Nature Neuroscience, I believe, which is so the ones I care about, this stuff you don't need to know about at all. I care about this right here. This is basically like a schematic, this is like a diagram, which is thought to explain how activity changes regarding the hippocampus and these other, it calls them cortical modules, but I just mean basically those outer cortical regions, depending on whether hours, meaning that long-term memory was freshly formed, or weeks, meaning the long-term memory was formed a long time ago, at least weeks ago. And what you're seeing here, these red lines are basically connections among these different brain regions. So you can imagine these are like different lobes, like frontal lobe, maybe parietal, occipital, or maybe a different part of the temporal lobe. These are different lobes of the cortex. This right here specifically is the hippocampus. And when you have a freshly formed long-term memory that's only like hours or maybe a couple days old, I kind of think of the hippocampus as kind of like a conductor, and that the hippocampus wants all of these regions to be able to communicate with each other, but there aren't a lot of connections formed among these different cortical modules. So what the hippocampus has to do when the long-term memory is freshly formed is it has to kind of send out messages to all of these different cortical regions, and then those cortical regions send back messages back to the hippocampus. And so like basically there's a network communication occurs, but the hippocampus is kind of like the conductor. It's organizing all of the communication among these different things. Like these regions are able to communicate with each other, but only once they send their information to the hippocampus. And the hippocampus is kind of collecting all of the different messages from these different cortical regions and kind of it processes them by itself. So the hippocampus is really important in this situation. To remind you, this situation is when the memory is freshly formed. The hippocampus has to organize like a conductor all of the different activities in these cortical regions because there's not a lot of connections among those cortical regions. However, according to this theory, after consolidation has occurred, the hippocampus isn't as important because you can see here now there are a lot of pathways that have formed between these cortical modules. Oops. Whereas previously there were no connections among them, now there is, and so you don't need the hippocampus to be the conductor anymore. So some memory that you formed like in your childhood or weeks, months ago, I can retrieve that information with just these regions right here because now there are connections among them. That's what consolidation in theory basically is. Those cortical regions form connections amongst each other over time, whereas when the memory is fresh, there are no connections among those cortical regions. But after weeks and weeks, once consolidation occurs, connections form, and the hippocampus is no longer necessary because you can retrieve the memory using those connections that have formed among those cortical regions. All right, that's it for chapter 9 on memory. So you can get out of here. I think I have meetings with 1 or 2 students. I'm happy to meet with those students now. I will stop recording.